I will, yeah. Before introducing our last speaker of this first part of the seminars, of the series of seminars, we are happy to announce that we will have a second part starting next month on the 14th of April, as you can see in the screen. And for the moment, we have 5 excellent speakers that have already confirmed, and they will contribute with new talks. And in particular, we will have the pleasure to count on Christian Schmeisser from the University of Vienna, Aitan Tarmo from the University of Maryland, River Sticky from Paris, Martina Conte from the University of Granada, and finally Lorenzo Pareschi from the University of Ferrara. So we will be happy to have you there for this second part. So I think I will stop sharing. I will let Philippe Maini share his screen, and in the meanwhile, I'll introduce him. So please, you can connect if you want. So today I'm pleased to introduce Philippe Maini. Professor Philippe Maini is a widely recognized expert in mathematical biology, and he's the director of the Wolfson Center for Mathematical Biology at the University of Oxford. Although he has developed most of his career in Oxford, in the past he has also enjoyed some positions at other universities like the University of Utah, Hokkaido University in Japan, the University of Electronic Science and Technology in China, Queensland University of Technology in Australia, or even the African Institute for Mathematical Science. So currently he is interested in the mathematical modeling of tumors with unhealing and pattern formation in early development, along with the theoretical analysis of the corresponding mathematical model which arise in these applications. He has around 300 publications in this field, and he has been awarded with several prizes like the Bellman Prize or the Arthur Winfrey Prize from the Society for Mathematical Biology. And well, today he will talk about modeling collective cell movement in biology and medicine. And please, you're welcome to start whenever you want.
Okay, well, thank you. So hopefully you can hear me. So thank you, David, for that very kind introduction. And thank you also for the invitation. And I should apologize. I think some of you will have heard a lot of what I'm going to talk about, but there are some new results sort of near the end. And so just see if we can get this to some problems happening there. Sorry, let's start with this one. See if this works. Okay, so hopefully that'll work. Okay, so collective cell migration is something that is a very common phenomenon. It occurs in normal development because in many cases cells are sort of born in one place, but then they need to migrate to somewhere else. Then in repair, in a wound, cells need to migrate to where the gap is in the tissue to help close it, and also in disease, where you actually don't want cells to move, but they do, and that leads to things like cancer, for example. And of course, I should mention that there's also a lot of collective organism migration. So for example, animals like bees, locusts, etc. So the outline of this talk, I'm going to look at 2 examples of collective movement. One example is actually not really angiogenesis, which is the formation of new blood vessels. It's sort of inspired by angiogenesis. And this is more a sort of mathematical, theoretical study. And then the second one is going to be cranial neural crest migration. And this is going to be more integrated with the biology. So angiogenesis, so basically what happens is that in wound healing and also in cancer, new blood vessels form due to cells called endothelial cells escaping from existing blood vessels and moving towards the wound or towards the tumor. And this process is called angiogenesis. And here is a cartoon of angiogenesis in the context of tumor growth. So a tumor secretes growth factors, these growth factors here, VEGF, FGF, etc. They diffuse to the existing vasculature, and then biochemical interactions occur, and these have been modeled in a lot of detail. And the result of those biochemical interactions is that cells peel away from the blood vessel, and they start to migrate up the gradient of VEGF, or whatever the growth factor is. And then they form vasculature. So there are many, many sophisticated models for angiogenesis. And we're not, and many, many people have worked on this, including ourselves, but we're not going to really talk about detailed angiogenesis. We're going to look at the simplest model and ask a fundamental question in mathematical modeling in biology that many people are working on, which is that if we propose rules of behavior at the cell level, how do we scale up to the tissue level? And one particular aspect of this is how do the parameters at the cell level relate to the parameters in the tissue level? And we're going to look at this first model that was proposed for angiogenesis a very, very long time ago. So the idea here is you've got the source of the tumor angiogenesis factor, which was VEGF or whatever you want to call it. And then you've got the blood vessels here. And what you see happening here is a phenomenon that's really a leader-follower phenomenon, where you've got cells coming off here, moving along. So the leaders are called tip cells. They respond to the chemical signal and do a biased random walk. And then behind them are followers or stalk cells. In fact, these are called endothelial cells that form the track, the trail. And then these trails can intersect with each other in a process called anastomosis, and this leads to a loop, vascular loop, and then the blood can flow around that loop. And the tip cells, the leaders, can bifurcate, they can branch. And this branching is thought to be related to some external signals. So basically what we're going to do now is we're going to consider a sort of phenomenon that's inspired by this example, which is that if you've got leaders moving in response to an external signal, and they are followed by followers, and the leaders can branch and they can merge in to other cells, how do you model that, given those rules at the cell level? And here's a classical model for it, proposed first in 1982 by Leah Edelstein. She's talking about fungus growth, and then various other people, including ourselves, have looked at this later on. So the idea here, and really, I'm sorry here, this should be little n and little e to be consistent with here. So here we have n, which are the tip, the leaders. So you've got diffusion, random diffusion, movement up the chemical gradient, where C is the chemical gradient, and so this is chemotaxis. Then branching, and it's thought that the branching is stimulated by the chemoattractant, so that's lambda NC. And then we have two terms here, which are loss terms. And this is that if a leader crashes into a follower, it'll form a loop, and so the leader will disappear. And if two leaders crash into each other, they will form a loop, so leaders will disappear. And these are anastomosis. And then the idea here is that this is the gradient of a flux flux. And it is the flux that is the source of the followers. So we just put the norm here, becomes a source term. And then typically a factor of 1 over h is put here to relate length to cell density, okay? And this factor here is something that I will look at very briefly. So one of the most famous models of angiogenesis is Anderson and Chaplin, over 20 years ago, where what they did was they took that Snailtrail model, they discretized it, added noise to it, and then they were able to show how, if you start off with some initial conditions of a few sources of the the leaders, as time increases, you start to get this vascular network. And you can see here, you're getting more branching because this is the source of the chemoattractant. So a lot of branching occurs here because the chemoattractant stimulates the branching. So what we're going to do is we're going to go from discrete to continuum. But before we do that, just something about the scaling factors. This is work that just appeared recently with graduate student Duncan Martinson and my colleague Helen Byrne, who many of you know. And the idea here is the following, that if we look at this flux term, so basically what's happening, if we think about modeling this on a grid, cells are moving along the grid, but the net flux here is what contributes to this term here, if we don't have the scaling factor in. But in fact, what could have happened is the cell could be moving back and forth, up and down, and each time it moves, some followers form. So it's creating vasculature while it's moving back and forth, up and down, but this here only looks at the net movement, so it underestimates the amount of vasculature formed. So if we think about this as being on a square grid, then we can look at the probability that you move up and down, the probability that you move left and right, work out the expectations, and then if we write these probabilities in terms of the movement will be a biased random walk, so we can write these probabilities in terms of the gradient of the chemoattractant, and then some straightforward analysis, which I won't go into, but which is in this paper, shows you that this kappa term should be of this form here. And so that is a derivation of this. In fact, this derivation has also occurred by other people in other contexts of this type of branching process. So now let's go back to discrete to continuum. And the important thing to point out is that in the Snailtrail model, the approach was the following. We looked at how individual cells moved, and we saw that individual cells do a biased random walk. We saw that as an individual tip moves, it stimulates follower production behind it. And then what we did was we said, let's just assume that the population does a biased random walk, and let's assume that the population of leaders leaves behind a population of followers. But what we're going to do here is we're going to actually take the rules at the individual level and systematically derive the density level equations and hope that we will recover the Snailtrail model that we've all been using. So we'll make life simple. We'll just assume that there's a fixed signal in one dimension. So that makes life very simple. And then we're on a square lattice. And therefore, if a cell is at lattice site ij, then the probability, if this was random walk, the probability that it moves in the positive x direction would be a quarter, and the negative would be a quarter, same for the y direction. But now we have a biased random walk, where that bias is stimulated or created by the chemical signal, and we just take a center difference here. And since the chemical signal is just in the x direction, the gy terms will be 0. So these will be 0, and we'll just have something here. And then we look at the anastomoses, and we represent these by functions, by reactions. So two tip cells collide, and they form endothelial cells. So that's represented by this. A tip cell collides with an endothelial cell, that's represented by this. Branching, we assume that the tip cell here, the leader here, branches like this. And that's just an arbitrary assumption. And the branching probability with the Snailtrail model, we put that proportional to the chemical concentration at that point. Okay, and then here, this is work of Samara Pillay, who was a graduate student of Helen Burns and myself. This is going back a few years now, and this is a simulation of that behavior. And what you can't see here, and I apologize for that, at the front, they're sort of colored black. Those are the leaders, and then the followers are in red. And then here is the source of the cells, and here's the source of the chemoattractant. And then as time goes on, we get it moved on to here. And then if we sum these up, what we see is we have a pulse of the leaders moving along in time, leaving behind, and then followed by a wave of followers, which is a traveling wave, but with not constant profile. Okay, and so what we want to do with this simple rule-based model is to develop a PDE from that. So we use a very standard technique. We do think about ensemble averages, and then we ask the question, how does the change in occupancy of ij, what contributes to the change in occupancy? So at iteration k, we've got a certain occupancy. At iteration k plus 1, we've got the new occupancy. What contributed to that change? So here, well, this looks very complicated, but it's very, very simple. The idea here is that, well, let's suppose the cell moved, so the probability of PM, and then this is saying that here's a cell, the probability that you're in a position I minus 1j, and that you moved to the right. Probability you're at I plus 1, and you move to the left. Probability you move up, probability you move down, etc. Those contribute to the occupancy in ij, but then you could move out, and that is the movement out. And then we have here tip-to-tip anastomosis, tip-to-sprout anastomosis, and the branching. So it's all very standard. And then the same thing for the change in occupancy of the followers, the endothelial cells. And then we do the usual thing that instead of being on a discrete lattice ij, we think of that as being this term here as being x minus delta x, or this term here, n at x minus delta x, comma y, and then we expand by Taylor, and we take the limits, and here's what we finish up with. So here's our equation here for n, and here are the limits. And so, for example, if we look at this diffusion term here, I'll mention more details of this later on, but if you just look at the diffusion term here, this is the standard thing here, where h is that the spatial discretization, so this normally you'd call this delta x. This is the time step, delta t. So this is delta x squared over delta t, and the usual thing that in the limit of delta t going to 0 and delta x going to 0, if this limit holds, is finite, and is d, then you get the diffusion. And so we just do that for all the other ones. The only thing to point out here is that we have certain processes here that the limit as tau goes to 0 of this process here would be infinite. But this has been looked at by many people beforehand. We're not the only ones to look at this. And the idea is, well, since in our simulations, we don't actually let delta t go to 0, we sort of assume that these things are finite. But that's really a fudge, something that needs to be looked at. The boundary condition 0 flux, that's all standard. And then if we want to get a one-dimensional model, what we want to do is we want to integrate across the y direction. We do that, and here is the model that we got versus the Snailtrail model that we've all been using. So let's compare the two. So let's look at these, the reaction terms, lambda nc, it's the same, the ne term, the n squared terms, they're all the same. But notice that here, the parameters we get in terms of the individual parameters. These are parameters to do with the individual movements, and I haven't really described those in detail, but they come from looking at how the probability of movement and the probability of interactions. And now look at this term here and this term here. It's the same as this term. The only thing is we've multiplied it by something here, which isn't here. Now, we've rescaled everything so n and e lie between 0 and 1. an and ae are parameters that lie between 0 and 1. So we could sort of argue, well, maybe this could be two numbers less than 1 multiplied together, two numbers less than 1 multiplied together. Maybe these are small if we're lucky. And so maybe this is 1, roughly speaking. And so we've, roughly speaking, got the same thing. Okay, so that's very, very wishy-washy and not very rigorous, but we're really trying to see, can we recover the old model? But now look at the equation for E, and that's just a completely different equation. Look at it, completely different. So this systematic derivation of the Snailtrail model has resulted in a model that's not the Snailtrail model. It's not the classical model. And I must say, that's not what we expected. One thing I should point out is that when we derived this model, in order to get something that would look like the classical Snailtrail model, we had to do things like ignore spatial correlation and make mean field approximations. So now we have three models. We have the cellular automaton model, which is the actual Snailtrail model. It's based on the rules, the actual rules. We then have the continuum model that we've all been using, which will be in red. And then we have this new model that we've developed, which is much more complicated, and that'll be in blue. So let's compare them all. So here's an example. So here are the N is the leaders, the tips, and the black here as time increases is the density of the leaders. Moves like this, and here's the followers. In red is the classical Snailtrail model. It lies on top of the black one, lies almost on top. And the new model, blue, lies on top, So perfect capture of the discrete phenomenon. But of course, two things to point out here. Firstly, these two very different PDEs are giving the same answer. That seems a bit strange. But secondly, if they're giving the same answer, use the simple model, which is the classical Snailtrail model. Now, here's another example. And now you can see there's a little bit of difference, but by and large, it's all the same. But once again, notice the blue and the red lines are on top of each other. In other words, the two PDE models, completely different, are giving the same answer. And now here's an example where we chose the discrete simulation so that we would get the wrong answer. Because we know that in our derivation of our PDE model, we assumed there were no spatial correlations. So what we did here was, for our initial setup for the discrete model, we made it very spatially correlated. And therefore, what happens is the discrete model, the leaders start off like this, the tips start off like this, and then they go to 0. The followers start off like this, and then they go to 0. But the two PDE models, again, they sit right on top of each other, and they both are wrong. Now, the important thing about this is that if, for example, this was experimental data, and this was your Snailtrail model prediction, you would say our model shows that the phenomenon we're looking at is not a snail trail. And that would be wrong, because this is data from a snail trail. We've simulated a snail trail to get these data. So in fact, the reason why the data don't match the model is because these PDEs are not the correct model of the snail trail. And because we've derived this model systematically, we know in advance it's not going to be correct, because we designed this simulation to make things really spatially correlated, and so that the mean field approximation was not valid. So we know it'll not work, but in the original model, these assumptions were all implicit. We didn't state them in the original model. But now we've stated them, we know why it doesn't work. Okay, so what we've done is seen that very different looking PDEs give very similar results for the cases that we studied, for the cases Samara studied. And the mean field assumption is where things break down. So very recently, Duncan Martinson, with her colleague Kazu, Ninumaya in Japan, Helen and myself, we looked at these two PDE models in order to ask the question, how come these two very different models are giving the same answer a lot of the time? And what we found was we identified two parameter groupings. And if these parameter groupings are small, we can show that the two PDE models at leading order are identical to each other. And so they're the same. So what are these parameters? Well, if you look at this first parameter, diffusion of the tips, branching rate divided by chemotaxis squared must be small. So if you think of the two parameters at the top, they're to do with movement away from a sort of two-dimensional movement away from the axes joining source of signal and source of cells. Chemotaxis coefficient is related, the bigger that is, the more is the biased walk. So it's almost like here we have a sort of fight between processes that are trying to diverge you away versus the process that's trying to move you along. And the idea is that if the process is trying to move you in the direction, one-dimensional direction, is the dominant one, then epsilon be small. And the same thing here, branching rate, which is pushing the thing apart, versus tip to tip, which is pushing things together. So intuitively, this makes sense, because remember, the original Snailtrail model said we're looking at how we know that an individual cell moves in a biased random walk, and we know that it leaves behind followers. So we're going to assume that the whole density of leaders does a biased random walk, and the whole density of followers is left behind. And now you can think sort of intuitively that if the dominant processes are concentrated in moving along in this biased random walk, then to replace cell by cell density is fine. But as soon as you start having the movement being two-dimensional, you can't simply say that the rules that a cell follows, I can just replace and say the cell density follows those rules. So the conclusion of this bit of talk is that incorporating the detailed microscopic behavior at the cell level. Now, I mentioned we made some arbitrary choices about the rules, like the branching. Had we made different choices, we would have got different nonlinear models at the macroscopic level. And so a key challenge is to understand how sensitive is the macroscopic level behavior to the assumptions made on the microscopic properties. And I showed that with two examples, the classical Snailtrail model and the one we developed. And I've shown how under certain conditions, they're both the same, but under other conditions, one is invalid and the other one is valid. And this is a key sort of problem at the moment in the field. How do you relate the microscopic to the macroscopic? And how do different rules at the microscopic, do they really make any difference at the macroscopic? That's still an open question. And so another key thing is that by systematically deriving the PDE model, we see what implicit assumptions were made on deriving the model, the Snailtrail model. So that was a more sort of mathematical modeling type of thing. So now for the second part of the talk, I'm now going to look at another phenomenon of leader-follower behavior, but this is going to be more experimental work, more related to experimental work. And this is cranial neural crest cell migration. And this is work done with experimental colleagues Paul Kolesa and Rebecca McLennan, who are at the Stowers Institute for Medical Research, my colleague Ruth Baker here, and a few generations of graduate students, Louise Dyson, who's now faculty at Warwick, Lana Schumacher, who's now faculty at Edinburgh, and Raza Ginionati, who's just waiting to have her thesis examined, and she's now working at university in Latvia. So what is a neural crest? So neural crest, these are transient embryonic structures that give rise to a lot of other structures in your body. So there are 4 neural crests: cranial, trunk, enteric nervous system, and cardiac. We're going to look at cranial, so one we're looking at. And I should mention, huge, just like in angiogenesis, a huge amount of work's been done on this. So here's again a schematic. Think of this as being the head, and this is the tail. This is the backbone, the neural tube, and the cells come out of here and they migrate as streams. Looking at another way of looking at it, here's the epidermis, here's the neural tube, and here are the cells coming out. And so we're looking at cells coming out of rhombomer 4, and they're going to branchial arch 2. And this corresponds to just below the jaw. So you can see here, we have a curved domain, it's 3-dimensional, and it's growing. Here is image, head, tail, rhombomer 4, cells coming out, these yellow things are cells, and they're moving to branchial arch 2. This is a cell, and here's the group of cells, and they're moving in streams as a collective. Why should we study cranial neural crest? Well, there are 66 birth defects identified that are a result of neural crest problems. In fact, cranial neural crest are very similar to the behavior of highly aggressive tumors, such as melanoma and neuroblastoma. And key thing is the cranial neural crest is experimentally tractable and serves as a powerful paradigm for collective cell migration. And we mentioned some of that in this paper here. So some more images, so our colleagues can follow the neural crest cells as they move along, they can follow them move along here. This is the length of the pathway, changes from, roughly speaking, 300 microns to 1300 microns, roughly speaking, in a day. This is just measuring the length. And it grows like this. So, important thing with the cranial neural crest, unlike other neural crest, there's very little proliferation of the neural crest cells during this migration process. So what we're going to do, if we go back here, this is curved surface. So we're going to take it to be flat as an approximation, no curvature. Moreover, we're going to assume that since this dimension is not very large, we're going to assume that it's a rectangle. So basically what we have here is a rectangle that's growing like this in the x direction, and cells are coming in at the origin, and as this domain grows, they've got to reach the other side, and they've got to do it inside a day. So a very simple calculation can show that random movement is too slow. So there has to be a directed movement. And it's known that VEGF, the same growth factor that's involved in angiogenesis, it's produced by the overlying tissue, and it's known that the cranial neural crest cells consume VEGF and move up the gradient. So the hypothesis is that this cell-induced gradient gives you the invasion. So we decided to test that hypothesis. So we start off with a reaction diffusion simple reaction diffusion equation for VEGF. So here's VEGF, C. It's moving in the domain has got length L. So we rescale to be 0, 1 in the x direction. So that brings in a 1 over L squared. And this is just a function L that fits. We just fit this curve to that growth curve. Here we take a simple production term, and this dilution term comes out of deriving the diffusion equation on a growing domain. We're going to take the cells as being units. So this is an off-lattice model. So a cell at center ij, xi, xj, xi, yj, yi, I should say, cell at center xi, yi consumes VEGF, and we're going to assume that it does it in a sort of Gaussian distribution. The form of this is not really important. Then we need boundary conditions. So we began by using homogeneous Dirichlet boundary conditions. Later on, we used zero flux conditions. And in fact, it's known in Xenopus, there's a chemical that's been found in Xenopus that causes a zero flux boundary condition. We're talking about chick. We don't know if the chemical is there. That's one of the open questions. In fact, what we found was that whether we used homogeneous Dirichlet or zero flux, it didn't make any difference for what we were looking at. We assume that the cells are discrete entities. They don't overlap, so there's volume exclusion. They randomly stick out a Philopodia, sense the gradient, and if the gradient, where they sense a VEGF, if that is larger than the VEGF concentration at the center of the cell will move in that direction with constant speed. So now we test a hypothesis that the chemoattractant produced by the overlying ectoderm is that sufficient for robust invasion. And I won't show the video, I'll just show the result. The answer is no. The domain here, it's growing, it's got lots of VEGF, the cells move in, they're sensing the VEGF, and they're using up the VEGF, and they use up all the VEGF, and now there's no signal for these cells, and they're left behind. In fact, biologically, it's observed that as the domain grows, the VEGF signal moves away from the neural tube. So this is consistent with the experimental observations. And the crucial thing here is the VEGF is too localized. The gradient is too localized. And so followers coming in later on, or the cells coming in later on, they can't sense the gradient. So what we did, Louise Dyson, Ruth Baker, and myself, was ask the question, what could these cells be sensing? And we said, well, suppose these cells sensed the cells in front of them and stuck to them. What would happen then? So in other words, let's assume that there are two cell types, leaders that move up the chemical gradient, and then followers that follow the leaders by sticking to them. Then we can get the invasion. So then we went back to the biology colleagues and we asked them, are all the cells the same? Because we think there's a difference in the cells. And they said, we don't know, but we will do some bioinformatics. So they took the trail they split up into 4 regions, and they did genetic analysis. And when you look at genetic analysis, you see there's differences. There's differences along here. So these cells at the front are different to the cells at the back. So we've shown that a single chemotactic gradient with a single cell type will not work. There must be at least 2 cell types. In fact, what they then found was there are differences between the cells at the front and the cells at the back. The cells at the front upregulate cell guidance and navigation genes. In fact, a lot of these genes that they upregulate are precisely the same genes that cancer cells upregulate. And what they found was trillion cells upregulate cadherin 11, and that's to do with the stickiness of the cell, makes it more sticky. So we've then used this model to go through a number of validations, experimental validations, and you can see this work is going back now almost for 10 years, been working on this problem. So here's an example of what would happen if you remove the followers. The model predicts the leaders will stretch out, and that's what's observed here. So the blue are normal, and then the red is the experiment, and the leaders that were normally here in the blue are being spread out. What happens if you take cells from the back, the followers, and stick them to the front? What our model predicts is incorrect. So what you see here is the cells that were put in the front, they actively migrate, whereas our model predicts they will just be advected. So the mathematical model here, it worked for the previous experiment, it doesn't work for this experiment. And I think you learn more when the experiment disagrees with the model. So then we said there must be phenotypic switching. These followers change to leaders. So the conclusion of that work was in high VEGF, followers become leaders. So a natural experiment is what would happen if you put VEGF along the trail. Then what happens, the model would predict, is that the followers, when they see high VEGF, they become leaders and they'll move up the gradient of VEGF. And so it'll split the stream into 2. These are cells following the original VEGF, and these cells follow the new VEGF. And this is what you get experimentally. The trail breaks up. So then Paul asked the question, how many leaders do we need for successful cell invasion? Well, we can't answer such a question because this would require us to really know the parameters very precisely. But we can give a qualitative answer. So we can ask the question, we can answer the question, what would happen if you increased the number of leaders? So here's the numerical simulations, and I'll just show the results here. This is normal. Here's the number of cells, and you get invasion. Here's the model prediction of what would happen if you increase the number of leaders. You get invasion, but a smaller number. Here's the experiment, here's normal, and here's the experiment. When you increase the number of leaders, you get invasion, but they're smaller. But notice the quantitative numbers are not the same. So we can't have a quantitative prediction, but we can have a qualitative prediction. So now I'll come to some open questions. How exactly are the leaders and followers communicating? Well, we suggested they're sticking to each other, but there's probably more than one form of communication. Could the leaders be secreting a chemical that's signaling to the followers? Could there be a mechanical interaction by contact guidance? Or could there be tunneling? And in fact, recently, our colleagues found that the leaders upregulate this water channel called aquaporin. And that stabilizes the filopodia, so it gives the cells a polarity and it creates tunnels. And so what they did here in this movie was, here is the control, and then here they inhibited aquaporin, and here they overexpressed aquaporin. So you see, when they overexpressed aquaporin, the cells are more polarized and they invade more. When they inhibited aquaporin, the cells are more sort of randomly moving, and then this is the control compared to the control. So what Raza did in her model was think about, remember we had the cells were sticking out filopodia to sense the chemical gradient. And now what we say is that when they stick out the filopodia, that filopodia in aquaporin overexpression stays fixed for a while. It's polarized for a while and creates a tunnel. Whereas here, it doesn't stay so long, and so it isn't so polarized. So then if we run these simulations, you can see here, so here the black are the leaders, the yellow are the followers, and you can see that in the gain of function, you're getting a lot of movement very quickly, and here you're getting not so much invasion as you're getting here. So qualitatively represents what's going on here. So there are other questions. Remember, I said that there were, we put zero flux boundary conditions. Well, why are we justified in doing that? In fact, our colleagues found something called DAN, which is an inhibitor of movement that's expressed partially along the domain, but is not fully expressed. We talked about, are there only two cell types? We mentioned two cell types. In fact, it turns out, and Linus Schumacher's done some work showing that, in fact, you can have more than two cell types, and you can get this movement to occur. And we assumed that the domain growth was uniform in space. It actually turns out, experiments show that domain growth is not uniform in space. And what this paper does here is explores then of how do we develop the model when there's non-uniform growth in space, and how do we use the model to make predictions. So what you can see there is that there's still a lot of open questions. But what this has in common with the previous work is that we have leaders and followers. In the previous work, it was tips and stalk cells. We have phenotypic switching, and I didn't mention, but in the angiogenesis, tip and stalk cells can switch. In fact, also what happens in the neural crest, I don't have time to go into this, is that the DAN is also expressed partly within the movement region to slow the leaders so the leaders don't get too far away. And then we've got this growing domain. And what we think is happening is that the cells must be stimulating or inhibiting the domain growth in order to control everything. So it's quite complex. You've got different cell types. They're changing their phenotype. They're changing their velocity so they don't get too far away. And they are interacting with the domain and affecting the growth profile. And finally, the phenomenon of leaders and followers occurs all over the place. I've given two examples, the example of angiogenesis, the example of cranial neural crest. It also actually occurs in tumor dynamics, in tumor cells that are metastasizing. They're leaders and followers. In animal movement, they're leaders and followers. In social sciences, they're leaders and followers. And so some of the ideas that I mentioned towards the end in terms of what are the key next challenges that we see in this area are summarized in these review articles. This one here in developmental dynamics is more from an experimental point of view. What are the key scientific questions? And this one here in the Journal of Mathematical Biology are more from the point of view of what are the key modeling questions. So I'll end there and thank you for your attention.
All right. Thanks a lot, Philip, for this very didactic talk.
All right, thank you.
Just that we have some time for questions. So people in the audience, either write down something in the chat or just turn your microphone on and ask whatever you think is sensible.
May I put a question, Philip?
Yep.
Thank you, very interesting and very communicative as usual from you. This is one of my problems when people get too technically into biology. I cannot follow.
Yes.
But with you, it's possible. Thank you. Actually, the problem of leaders and followers, as you mentioned in the very last slides, is very common in very many animal. And the class of systems have been examined mainly in swarm theory. It's very tricky. It's very tricky. And what I have found is, a general indication, which probably is also valid in your own biological system, but this is what I wanted to know. The way the followers follow the leaders depends not only on individual mechanical conditions, say in position, velocity, and say distance, which is a consequence of, but also on the local density conditions and local gradients. Does it happen also for cells?
Yeah, so here, I didn't mention too much detail, but we also have here volume exclusion. So
yes. One of the cases, yes.
Yeah, so that it also would depend on the densities, and also the number of leaders and the number of followers then also depends on cells are trying to come in from off the neural tube and come in, but they can only come in when there's a space available for them to move in. And then depending on the concentration of the signaling chemical, then that will determine whether you're a leader or a follower. So there's two signals already there going on, but certainly the density plays an important role in this one, we think.
In that sense. Thank you, Philip.
Okay. And this important thing, Nicola, I should have mentioned that, of course, you've done a lot of work using kinetic theory, addressing these issues, which is a different approach to the one that we've used. So there are many different approaches to looking at this problem.
Not necessarily useful, that is.
Well, we don't know in this.
Thank you.
We have a question by Francesco Cavalieri. Francesco, if you will.
Yes, thank you. I want to ask about, in a certain point, we say we take a one-dimensional model of the Snailtrail model, but I can figure how this one-dimensional model have the same behavior, because the different heads can meet each other in a very different way in one dimension. I understand correct that we consider it a one-dimensional model or not?
Well, so the original idea of the Snailtrail model was that it was somehow looking at doing the thing in 2 dimensions, and then looking at the cell density. So, for example, the cell density along 1 dimension. So the cell density at a particular x value would be adding up the density of all the cells in the y direction. And that was the idea of the original Snailtrail model. So that you were almost like summing up all these different processes that were going, and then you were projecting them onto a one-dimensional process. Because obviously you're right. I mean, if everything's moving in 1 dimension, then how these things can interact with each other and collide with each other is very different to what would happen if you did it in 2D. So the original model was that really think of it in 2D, but then we're sort of averaging to get it into 1D. That's what the original model was looking at. And then what our recent work suggests is that is reasonable if, by and large, all the action is happening in 1D. But as soon as you get a significant amount of action happening in the 2nd dimension, then it's not a good approximation. So it's exactly what you say. Yeah, it can't, as soon as there's a lot of things of branching and things like that happening in the 2nd dimension, then as we would expect intuitively, this one-dimensional description is not a good one.
Okay.
Okay, I also have a question myself, which is related to one of the last numerical simulations you did display about the neural crest migration. So it seemed to me that those cells which had the enhanced phenotype, they were sort of creating subclusters along the migration, or maybe it's just a...
No, yeah, that's right, because you're absolutely right there, Juan. What they were doing, what the colleagues found was that these cells are creating a tunnel through the tissue that they're moving. And I didn't explain this properly. So in our model, what we have is that the followers, if they get into a tunnel, then because the tunnel is sort of like a hole, they're more likely to move in the tunnel. They are outside the tunnel. Now, there's still the question that once you get into the tunnel, how do you know what the right direction is? You know, to move in that direction, not in that direction. Partly, you know, if there are other cells already there, then by volume exclusion, you'll move in this direction. But that's still an open question. What is the signal that makes you move in the correct direction of the tunnel? And could it be that the creation of the tunnel and maybe also the growth of the domain is sort of aligning things in a certain way that makes you move? Yeah, so I should have explained that properly. We then, as the leader is moving along, it creates a track on which it's easier to move. And that's why you get these streams moving along.
Okay, I see. Thank you, Philip.
Also, I have a question.
Okay. Yeah.
Do you mention that, for example, BGF, like chemoattractant for the angiogenesis, and also you mentioned apoptosis. Who molecules are involved in this process? In this process of in particular in angiogenesis.
So in angiogenesis, I actually don't. I mean, there's been a lot of work done in haptotaxis. Like people like Mark Chaplin, for example, done a lot of work. I'm actually not sure in what those are. I think they're probably the same as in the NeuroCrest. In the NeuroCrest, there's cadherins that are involved in the haptotaxis, and I would assume that they probably also play a role in the angiogenesis, because what we're finding is that a lot of there's also biochemical things like Ang1 and Ang2 that play a big role in angiogenesis. And we find they also play a big role in NeuroCrest. So I know that definitely the NeuroCrest, the cadherins are playing the role of haptotaxis, are one of the haptotactic signals. So it could be that they're also for the chemotaxis, for the angiogenesis, but I'm not sure of that. Yeah, I would have to look that up.
All right. It seems that there are no more questions, at least not in the chat. So if it's okay, we may close this seminar here. So thank you again, Philip, for the talk and the discussion, and everybody for attending. And I don't know, Juan or David, would you like to make some final statement or announcement? Or shall we end here?
No, we'll not only we will send the specific like in David's presentation, specific conference after in April, after this holidays period, and we will send a message to everybody. Okay.
Yeah, I guess we will also upload to the website very soon, no? So it will be available for everybody. But yeah, we will send some email.
And also to thanks Philip for the real interesting.
Oh well, thank you, and thank you for the invitation. And it's good to see.
Thank you to accept this.
It's good to see you in good shape.
Well, thank you. And then it's good to see lots of friendly faces again, and hopefully we'll meet in person soon. Be very good.
Hopefully so, yes.
Yeah.
So, meanwhile, I would say everybody enjoy your Easter break and hope to see you again in the second round.
Yeah. Okay. Take care. All the
Thanks,
best.
everybody.
Bye.
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 Let's finish the recording. I can't find it.